Good morning, everyone. This is September 4th, 2018, Microbial Minutes, the American Society for Microbiology's every couple of week update on what's hot in the microbial sciences. I'm Julie Wolf, the Science Communication Specialist here at ASM, and we're going to talk about several hot topics that have been making waves in the last couple of weeks since our last session. So the things that we'll be talking about today include a foodborne E. coli that has been linked to UTI, urinary tract infections, that are caused also by E. coli. We'll be talking about a new bacterial tree of life. Then we'll move into some reports from the CDC on HPV trends, vaccination, and cancers concerning this virus. And then finally, we'll end with some new anti-Ebola antibody reports and a little bit of update on the outbreak that's ongoing in the Democratic Republic of the Congo right now. So let's dive in. We're going to start with a report from ASM's MBio journal that's titled Escherichia coli ST131-H22 as a foodborne uropathogen. So the take-home message from this is that foodborne bacteria are linked to UTI causing E. coli. And this is a big finding because although there have been suggestions of the things that we eat contributing to the E. coli that can cause UTIs, there hasn't really been a very strong link. This is one of the strongest links in this report here. So let's talk a little bit about E. coli. It comes in many different flavors, which are called serotypes. Serotypes is basically determined by the immune system interaction with the outside of the E. coli, often with the different components of its LPS or other parts of its outside envelope. But it's basically how it looks to the immune system. And the serotype called ST131 is a particularly bad serotype. It's associated with a lot of genes that aid in its virulence. It's resistant to some pretty important antibiotics such as ciprofloxacin, and it has a beta-lactamase that can chew up most of the penicillin related compounds, or it just makes it a really recalcitrant infection, one that's hard to treat, in addition to one that can cause some pretty nasty disease symptoms. This is the most common extraintestinal pathogen, meaning that it's extraintestinal. It's often found as part of the natural commensal bacterial of bacterial composition of an individual, and then under the right circumstances, it can cause disease, right? So things such as a UTI that can be linked with the different microbiota that are associated with one's gut flora. So the question is, how does this really nasty E. coli serotype colonize people in the first place? And what I'm showing you here is the emergence of ST131, which hasn't been around for that long. I mean, it only emerged as a major pathogen in 2002. So if you look here, this ST131, there's this tiny little dot back here where it was not really something that was of major clinical concern. These are different representatives of serotypes that are proportions of clinical isolates. You can see that it quickly established itself as one of the major serotypes causing disease. So how does it colonize people? That's what this study was aiming to find out. This was a prospective study, meaning that rather than taking data that they already have, as you would do in a retrospective study, they set out to collect different isolates of E. coli from several different sources. The researchers involved in this study, of course, examined the isolates from different clinical isolates, those that were collected from people who were sick with E. coli. And they also went around to different grocery stores collecting meat and then testing the meat products for different types of E. coli. Now, having different types of E. coli associated with meat is not too surprising. There's natural E. colis that live among almost all different types of organisms, especially mammals, which if you consider like pork or beef, that's going to come from a mammal, but also with, excuse me, with poultry, things such as chicken or other more exotic poultry such as ducks or geese. And so they took these different isolates that they were able to find, and then they looked at their the characteristics of these by molecular means. They did multi-locus sequence typing. So they looked at what that sequence type or the serotype was, but they also looked at the sequence type. This is looking at some common genes that are often found in different E. coli isolates. And they also specifically looked at FIMH, which is a gene that can be polymorphic and also used to categorize different E. coli lineages. They did core genome phylogenetic analyses, looking at some of those genes that are part of the very basic genome that are associated with many different E. coli isolates. So that would not necessarily be virulence factors, because not all E. coli will contain virulence factors, but some of those core genome properties. And then finally, they also looked at the presence or absence of a very specific plasmid, looking for something called the coli or col5 poultry-associated plasmid. And that's because this is a plasmid that facilitates growth of E. coli inside poultry. So what did they find? Well, first, as I mentioned, they found a lot of E. coli on those meat isolates. We probably shouldn't be too surprised, but 80% of the different meat isolates contained some sort of E. coli. Of course, there were hundreds of different serotypes. They were not all concentrated in those clinical serotypes, the type that can cause disease in humans. There were 27 of those 2,400 positive samples that did have the same serotype, that ST131, and a very specific lineage of ST131. So ST131 can be further divided by more specific molecular means, and they found that it was ST131-H22. And this is, you will see, is important in just a minute. But almost all of these were from poultry, and specifically chicken. So like 93% of these ST131-H22 were from chicken, and all but 2 of those isolates carried that plasmid, that specific plasmid that allowed them to colonize, to more efficiently colonize different poultry. So when they looked at the E. coli from patients, they took culture-positive clinical samples, so samples that they knew should grow E. coli, because they had already been diagnosed, the patients had already been diagnosed as having an E. coli infection. And from those, they had around 1,700 of these samples, and they found 72% of those they were able to isolate or re-isolate the E. coli. And this was not highlighted in any of the news stories, but I found it extremely striking that they were able to obtain more E. coli from the meat samples than from the culture positive clinical samples that had already been shown to grow E. coli. But that probably has to do with the amount of samples. Often you have a very small biopsy or swab that's taken from a patient, a human patient, compared to the bulk meat that you would buy in the grocery store. Now, the types of patients that they were looking for were not necessarily the types that you might imagine when you think of E. coli. You think E. coli, often you think GI distress, you think diarrhea. But a lot of the patients that they were examining had cystitis and pyelonephritis, meaning that they had more serious forms of UTIs. So a UTI, or urinary tract infection, can be a fairly benign infection if it's caught and treated and resolves quickly. But if it doesn't, if those things don't happen, it can move up that urogenital tract and go into some of the internal organs that produce the urine that will come out through the urinary tract, things like the kidneys. And so you can get a much more serious disease if the organism is able to reach some of those internal organs. And so especially within these particular group of patients, they found that there were 24 isolates of E. coli that were this very same strain that they had seen in the poultry, the ST131-H22. 6 of those were exactly the same as the meat isolates. And there's a little asterisk there, and that's because they did some very high resolution techniques. They did the core genome analyses, and they did all this MLST, but they did not do whole genome sequencing. So it is possible that there's a SNP, like a single nucleotide polymorphism, that might be able to differentiate some of those meat isolates from the patient isolates. But using what they did have, they were not able to distinguish those, and so it looks like they're identical. And those 6 meat isolates also carried the coli plasmids, suggesting that they were perhaps more familiar with a poultry or an avian environment than necessarily a mammalian environment. And so I'm showing you a figure from their paper that I find quite striking. This is a timeline, but it's a very interesting timeline because it's actually a concentric timeline. So all of the decades are shaded in gray, and you can see the evolution of these different lineages over time as demarked by the different lines here, right? And there's a lovely legend that you can see up here where you see the human clinical isolates are in yellow, the meat or animal isolates are in red. There are various indications of, for example, the coli plasmids, which have little circles like a circular piece of DNA that could be a plasmid, for example. And then you can see where those coli plasmids were acquired, and they identified 6 unique and independent acquisitions of those coli plasmids that allowed some of those isolates to become better adapted for growing in birds. Now, to see that resolution or the relationship between the clinical isolates, these 6 clinical isolates that were the same as the meat isolates, I think that this figure is particularly striking. So you'll see here that there's a number of different H's. There's H30, H27, H22, and H22 is the subgroup of the ST131 that we're talking about right now. If we take a zoom in here in panel B, you can see the relationship in this phylogenetic tree of those different isolates that came from chicken, turkey, pork, and you can see them lined up here, right? And you can see that the human clinical isolates seem to group on their own, away from those meat isolated E. coli samples. But if you look at within these groups of red where you see the meat samples, you can see that there are some human clinical samples that group more closely with these meat isolates than with those that were more likely to be associated with the human clinical groups. And so these are the ones that the science team suggested come directly from the meat samples themselves. And so this was quite a big finding because there have been a lot of studies looking at, for example, the lineage of H30 within ST131, and studies have not been able to support the transfer of these different human clinical samples that have come from these different meat contaminated meat sources. But this study does suggest that there is a crossover and that the cross goes in a direct direction, right? Because you imagine that there are farmers or animal handlers that are handling meat, and they also have E. coli. And it's possible that people could actually be the source contaminating the meat that is then handled by different animals, right? And could be passed around within an animal population that way. So the infection route is not unidirectional, it can actually be bidirectional. And so what this study suggests is that the direction is from fowl or from these poultry samples going into the clinical or the human population rather than vice versa. And so this was a big story. It hit a number of different locations, some mainstream, some not so much. UPI featured a press release titled E. coli strain in poultry may cause infection in humans, study says, with a quote that was from Lance Price, the lead author, the head scientist of this group, where he says, in the past, we could say that E. coli from people and poultry were related to one another. But with this study, we can more confidently say that the E. coli went from poultry to people and not vice versa, as I was just saying. They also featured a quote from the first author, the scientist Cindy Liu, in CIDRAP, a group that deals with infectious disease and resistance in University of Minnesota. And they had a very nice write-up interviewing her and other scientists involved with this study, and she said that this particular E. coli strain appears capable of thriving in poultry and causing disease in people. Poultry products could be an important vehicle for bacteria that can cause diseases other than diarrhea. And so in addition to featuring the directional transfer of E. coli from meat products into people, it also highlights the importance of E. coli as an organism that can cause diseases that are not only associated with Chipotle or other types of beef recalls, as you might frequently hear in the news, but also things that can cause other types of serious diseases, such as kidney disease through urinary tract infections. This study was shared by a wide variety of different individuals, including those that are involved with public health. Of course, people are interested in maintaining a clean chain from farm to table, and also by science reporters, and also by the meat industry themselves. And despite the fact that they do have a vested economic interest in maintaining production in the way that it's done, they did highlight most of the data correctly, even if some of the scientists representing the meat industry in this particular article were skeptical of the interpretation. Okay, we're going to move on and talk about our next article. This is from the journal Nature Biotechnology, titled "A Standardized Bacterial Taxonomy Based on Genome Phylogeny Substantially Revises the Tree of Life." And so the take-home message from this particular study is that the bacterial tree of life is reorganized based on gene orders. And I know that my first thought when I saw this article is, hold on, didn't we just get a new tree of life? There was a big study that came out a couple of years ago from Hug and Joel Banfield's lab, in which a new tree of life was shown that incorporated many unculturable or perhaps not yet able to be cultured bacterial species, including the candidate phyla radiation here, and then tying their relationship in with the eukaryotes, the archaea, with humans represented all the way down here. And this is a very nice image that has been highlighted in a number of different outlets and continues to be shared quite widely. However, it is important to continually revise things like our phylogenetic trees. Why is that? Well, not we, but scientists and the way that the public interprets what scientists find, use these sequence-based trees to provide a framework to develop taxonomy that will help to explain and account for the evolutionary relationships and the differing rates of evolution within different species. So many of the current tools that take different sequences and group different types of bacteria based on that may tend to group certain taxa more than others. And that's in part because there's not as much information about those different taxa or the different species that are, or even genera that are within those particular taxa. That's in part because we just have more detail about some certain well-studied taxa. There are some bacterial groups that we do not have as large a sampling of. And another reason is because many of these tools and many of the workflows rely on 16S ribosomal RNA gene sequencing, which is a good sequence and has certainly played a huge part in bacterial taxonomy, but can be somewhat limiting because this is only a few hundred to a few thousand base pairs that we're looking at. And when you imagine the way, the size of a bacterial genome, this is a very small part in it. And so what this group did was to not reorder the entire tree of life. I specifically in this summary said the bacterial tree of life. And this group looked specifically at the bacteria, this group in purple here, to see if they could refine the bacterial organization part of this tree. And they did that by looking at the order of genes. So they took concatenation of 120 ubiquitous single copy proteins. So concatenation means the order. So if we're looking at, say, 5 different samples and the samples have different genes A, B, C, they all have A, B, C, and 4 of them have that gene order A, B, C, and 1 of them has A, B, and then C is located somewhere else in the genome. Perhaps there's been a translocation, or maybe it was a transposon that hopped out and then hopped back in. You could make an assumption that 5th sample would actually be further related. It would probably have some sort of common ancestor because they both have A and B in the same order, but because C is no longer in that same order, it's not able to be grouped with that original 4 samples. And so they used this only instead of using A, B, and C. They had 120 different single copy proteins, so they didn't want to confuse this by looking at homologous genes, things that might swap out with one another, for example. And they use this to propose a new bacterial taxonomy, which you can see here illustrated on the right, shown in a couple different ways here with the relationship of genes shown in a spike-like fashion, and here the lines themselves are what represent the relationship with phylum, class, order, family, genus, and species represented by these different colors. Using their new method, they were able to propose a taxonomy including almost 95,000 bacterial genomes. And so here they're looking at genomes because they're looking at the gene order, right? They don't want to only look at the one gene sequence. And this really makes sense when one thinks about the way that sequencing projects are moving forward. It's much more common to look at a community of microorganisms by looking at all of the DNA sequences that are there and then using different workflows and pipelines to figure out the unique genomes based on a number of different references that we now have, and use that in order to see what the representative bacterial species are within that particular sample. They were not only able to use those very well-described taxa, but were even able to include 13,600 uncultured organisms, those whose only information we have is their gene sequence and not necessarily what their favorite agar food is. And so this was received with quite a bit of excitement. Donovan Parks was the first author, and he gave a great quote to phys.org, and you can actually find this. I think it was the press release from the University of Queensland, but it was republished in a number of places. Parks said that this tree helped us to create a standardized model where we fixed all of the misclassifications and made the evolutionary timelines between bacterial groups consistent. For example, the genus Clostridium has been a dumping ground for rod-shaped bacteria that produce spores inside their cells. So we reclassified this group into 121 separate genus groups across 29 different families. And so this is great. It will help people to better understand the relationship of different bacteria that have perhaps similar phenotypes, meaning that they look similar, but slightly different genotypes, so that their nucleic acid and their DNA sequences are different. As I said, this was well received. It's called an exciting paper, but as you can see here, there are some scientists who are now wondering, do we rename things officially? No? So confused. And it's true, reclassification can be very confusing. This has been the case in the clinical microbiology community, where Clostridium difficile has officially been renamed as Clostridiales difficile. So it's still called C. diff. And you see within some practitioners that it is being called Clostridiales, but it's still common to see in different publications the reference to Clostridium difficile, which just so happens to be the same genus that Dr. Parks mentioned has been a dumping ground and needs to be perhaps a little bit better refined. I'm sure that this will be a continuing area with more resolution coming up. If you are interested in bacterial taxonomy in general, I did want to highlight an article that was in the New York Times Magazine a few weeks ago that was an excerpt of an upcoming book from David Quammen that is about Carl Woese and the way that bacterial taxonomy was somewhat his contributions to bacterial taxonomy, and just a very fascinating read that I highly recommend if you're interested in the history of how bacterial classification was reorganized itself. And of course, we'll link to that down below the presentation window here. All right, we're going to move on. Our next discussion is going to be on two different morbidity and mortality weekly report articles. They were in the same issue, and so they really are quite related. One is titled Trends in Human Papillomavirus Associated Cancers, United States, 1999 to 2015. And the other one is titled National, Regional, State, and Selected Local Area Vaccination Coverage Among Adolescents Aged 13 to 17 Years, United States, 2017. So the take-home message from these studies is that broader HPV vaccination is needed to stem the spread of cancer. And not all cancer, of course. We're talking about HPV-associated cancers. HPV, particularly certain strains of HPV, are associated with the ability to mess with a cell's checkpoints. It's cell cycle checkpoints. So the ability to detect certain irregularities in DNA and the ability to continue replicating despite potential mutagens or mutations having occurred within the cell. And so those can lead to the accumulation of additional mutations that will eventually lead to the development of carcinoma. And so most of the cancers that are associated with HPV are cervical cancers. So cervical cancers are caused almost primarily by HPV infection, not 100%, but 99.9. But HPV is also associated with other types of cancers as well, and we'll talk about that in just a second. Now, these cancers are now preventable with the introduction of HPV vaccination. HPV vaccination was introduced in 2006. It's a series of vaccinations. It's not a one-time jab. You think you have to go in. There are two companies, last I checked, there are two companies that incorporate either 2 or 4 of those HPV strains into their vaccine mix, and you have to go in either twice or 3 times for boosters to ensure that you are fully protected from infection with the virus. And I believe, at least when they first came out, that it was recommended that women 26 and younger be eligible, at least for insurances, to cover the vaccination. That was eventually moved into also being suggested for men on account of this is often sexually acquired. And so if you can protect men, you might protect women, and also men are also susceptible to certain types of HPV-related cancers. And I think that now the recommendations even go higher than 26, but I would have to get back to you on that because I can't remember exactly. Anyways, there are a number of different campaigns to increase vaccination, but none has been more successful than in Australia. This was an article, this was research that was highlighted a few, I want to say months ago. We didn't talk about it here on Microbial Minutes, but it did make the news in a number of areas, talking about how in Australia, in particular, there's been a decrease in some of these cervical cancers, and also with a more quickly developing phenotype or disease symptom, which is genital warts, which can be also associated with some of these HPV strains. So if you look at this graph, you can see before the campaign and after the campaign that there is a drastic decrease, especially in young women who are targeted for this vaccination program. And so the question now is, how about the United States? The United States has not been as aggressively campaigning to get HPV vaccination coverage quite as broadly implemented. So we're going to first talk about the National, Regional, State, and Selected Local Area Vaccination Study. This is one of the graphs from that study. They looked at a number of different types of vaccination. They didn't only look at HPV, they looked at Tdap, which I believe is tetanus, diphtheria, and acellular pertussis. This is a booster to something that one should get when one is early years. You can see that coverage is quite good. They also looked at HPV, and they looked at HPV vaccination for whether people had more than one, whether they had one, whether they were up to date. So UTD is up to date. And what they found was that specifically for HPV vaccination, half of US teens are not fully vaccinated against HPV. They may have received a single dose, and in fact, in 2017, nearly 2/3 of these adolescents, 13 to 17-year-olds, did receive that first dose, but only 49% received the 2 or 3 doses necessary for full protection. So that again suggests that they might not be fully immunized against the virus should they encounter it later on in life. The researchers did find that on average, the percent of adolescents who start that HPV vaccine series grew 5% annually. So it's better than it was when the vaccine was introduced in 2006, but still has quite a ways to grow. All right, we're going to look at the next study, looking at the trends in the HPV-associated cancers. And here is a graph. It looks like a bunch of lines just running from right to left, but there are a few minor changes of a few percentage points. And so I want to highlight some of these. This research team found that cervical cancer rates are decreasing by 1.6%. That's promising. That suggests that it may be in part to changing sexual practices. It may also be in part because there is more HPV vaccine being administered to young women before they are sexually active and potentially exposed to this virus. Sidebar, let me just say that there are ways that you can be exposed to this virus that are not necessarily sexual. That's most commonly associated with being a sexually transmitted infection, but it is also something that can be acquired non-sexually, and yet another reason why one should get vaccinated, even if one is not sexually active. Anyways, anal cancers tied to HPV. So there are other non-HPV associated anal cancers, but those that are specifically associated with HPV did increase, however, 2.9% in women. I don't know why I have girls here, sorry, and 2.1% in boys. Similarly, there were changes in throat cancers that increased 0.8% in women and 2.7% in men. And so this suggests that although the overall cervical cancer rate may be decreasing, there are other associated cancers that can be tied to HPV infection, and those need to be monitored as well. All right, so this could be reported in a number of ways. And when looking at how different mainstream outlets reported this, I found this particularly interesting. If we look at the Yahoo Finance report, it's really glass half empty here. Vaccine tied to the drop in HPV cases, still not reaching half of US kids. That doesn't sound very promising. It sounds like, okay, well, we need to do more work. However, if we look at Time magazine, they presented a much different headline saying that more teenagers than ever are getting the HPV vaccine, says the CDC. Obviously, these things are both true. We just went over the data. The HPV vaccine is still not reaching half of the US kids. It's at 49%, but it's at a 5% annual increase, so more teenagers are getting the HPV vaccine than ever. Now, how this is presented might influence one's, the reader's view of what needs to be done. And so I particularly liked an Axios paper or an Axios article that was brought to my attention by Peter Hotez, who I believe I've mentioned in these sessions before. He's a vaccine advocate and neglected tropical disease researcher. He highlighted a piece by Ron DePino, who is at the University of Texas, who wrote a really great piece, and we'll link to that, of course, which highlighted that the bottom line is the 33,000 HPV cancers that will be diagnosed in the US this year reflect the lack of vaccination in the past decade. So it doesn't really matter whether you think about HPV vaccination as increasing or decreasing. The end result is that people are still coming down with cancers associated with this virus when, in fact, you can protect someone from infection with this virus in the first place. And so broadening the vaccine application seems like the best move. All right. We're going to move on to our last topic. This is one from Immunity. This is a report titled Multifunctional Pan-Ebola Virus Antibody Recognizes the Site of Broad Vulnerability on the Ebola Virus Glycoprotein. The take-home message from this is that an antibody protects against multiple Ebola virus species. This is a pretty big deal. So we know that Ebola continues to be a threat. There's the ongoing outbreak in the Democratic Republic of the Congo. We talked 2 sessions ago about how there are now 6 species, but that one that was just discovered is unknown about whether it causes disease in humans. It was found in bats. It can infect human cells in vitro, but has yet to cause any human disease in people. There are 3 species that we do know can cause human disease, and we know that because they've caused outbreaks. And that includes the Zaire Ebola virus, the Bundibugyo Ebola virus, and the Sudan Ebola virus. There are many different both vaccination and treatment suggestions and ongoing research aims that might eventually make Ebola not be such a serious threat. One of those, which you may remember hearing about around the time of the West African outbreak, is the ZMapp. ZMapp, which is a 3 antibody cocktail. So it requires 3 different antibodies, and it protects only against Zaire Ebola virus. All right, so it does not protect against Bundibugyo or Sudan species of this virus, and that makes it good in outbreaks like what's ongoing. The Zaire Ebola virus is the Ebola virus that has caused the most serious outbreaks, but it is not the only one that can cross and jump from an animal reservoir into people. And so it's important that we're able to ward off infection for all. And we'll talk a little bit more about ZMapp after we get through this study. So the question that this research team wanted to ask is whether protective antibodies could be isolated from Ebola patients that would be pan-protective, meaning protective against many different Ebola viral species. And be that broadly neutralizing antibodies that we often talk about in terms of HIV, because that can also present many different forms of the virus, but something that would be broadly neutralizing, able to ward off infection with a number of these different viral species. So what they did was to take the blood of Ebola survivors, people who had survived that 2014 to 2016 West African outbreak, and they screened 600 antibody fusion cell lines for the ability of the antibodies produced by those cell lines to stop infection in vitro. So all of this work was done with cells in petri dishes. They then did some structural work to look at how those antibodies interacted with the glycoprotein of Ebola virus. The glycoprotein is the outermost protein that is important first for uptake by endocytosis, and then for escape from the endosome after it's been taken up. And they identified two candidates, EBOV 520 and 515, which bound to the glycoprotein in a way that prevented infection of the cell. So they were neutralizing, largely worked by neutralization. And then they showed that both of these antibodies protected broadly against many different species in many different animal models. So let's look at some of that survival data. If we have here an A, B, C, and D, we're looking at a mouse, a guinea pig, and a ferret challenge with both a Sudan Ebola virus, and Bundibugyo Ebola virus. And remember, this is a virus that an antibody that they've already tested against Ebola virus, the Zaire Ebola virus, which is what the original patient had been infected with. And so you can see protection in mice using this 515 protects slightly better than the 520. They tested 520 in guinea pigs against the Sudan. Also saw protection here, not significant, should be noted. They looked at the Bundibugyo Ebola virus, and they could see not quite, it was not able to save even half of the rodents that they were looking at here in the ferret model, but they were able to get 25% saved, which is better than none. Most of them died after 8 days. And those that did survive had much lower viral titers than those that had been given a sham or antibody against dengue virus. And what they have here written is the IgG4. They identified an IgG4 from 520, and most of the time when broadly neutralizing antibodies are found, they are either an IgG1 or an IgG3. And so I just wanted to show you what the difference is here. The ability to bind is in this section of the antibody, and then the Fc receptor, the part that interacts with potential immune cells and can direct the cell's activity, is in this Fc part here. And so this is a highly glycosylated, but still protein section of the antibody. And what they were able to show is even if they substituted the IgG4 and they swapped it out for an IgG1, they still had protection. They, as I mentioned, showed that the antibodies worked primarily by neutralizing the virus, and they bound to epitopes that are different from those of other broadly neutralizing antibodies that have been identified and can work against other Ebola. They work across several different Ebola species, but they do not confer it's only partial protection. It's not entire protection. Of course, since this is in a rodent model, the next step is to look in primates, because there is not always 100% correlation between what is found with Ebola models in rodents and what is found with Ebola models in primates, which is the closest thing that we use to humans. And they also identified that some antibodies from their screen, which I'm not showing you here, acted synergistically, meaning that if they were added in combination, similar to that ZMapp antibody, they would even improve upon the protection that they saw here. So that leaves several different roads for this group to go down, looking at either a monoclonal antibody, which would be probably the ideal way to go, or perhaps some sort of combination therapy. So this was written up in a commentary where the commentary authors commissioned a very nice schematic looking at exactly how those 515 and 520 antibodies bound to the Ebola glycoprotein, this is the outside of the Ebola virus here. You can see that it's binding and not allowing it to escape the endosome. Even though it's taken up, it can no longer interact with the particular protein that it's necessary to interact with to escape that endosome. And so this was written up in a report from Vanderbilt where the researchers were located, and the senior scientist, James Crow, stated that the antibodies appear very promising for development as treatment and prevention for Ebola virus disease. Of course, if you can find an area of the virus that is good for conveying, for treating and preventing infection, that also does make it a good candidate for some sort of vaccine. If you can get the immune system to recognize this particular epitope before being exposed to Ebola, it's one way that you can prevent infection from occurring in the first place. This is similar to what is being used right now. So there is, as I mentioned, the ongoing outbreak in the Congo, and there is ring vaccination with a vaccine, which we have previously discussed in Microbial Minutes, that is ongoing. And I want to point out that this is not the only antibody that is showing promise against Ebola. There's a number of different Ebola antibody studies that are being published right now, especially because there's so much research coming from the number of survivors, and perhaps that one can find the different immune capabilities that allowed those survivors to survive a bout of Ebola, which could then confer protection on future potential outbreaks. So here, in May, the NIH announced that they would begin testing of a monoclonal antibody called MAB114. This is something that can be administered intravenously. They were doing this for a safety check. This is not to be effective, to actually test against individuals who are infected with Ebola. However, in August, I believe it was in August, Anthony Fauci of the NIH announced that the antibody was in fact being used in conjunction with other types of trial treatments during this ongoing outbreak, because people are very worried about the current Congo outbreak, on account of it's happening in a place where there's also a lot of conflict, and access to patients is not very thorough. And so it's possible that, for example, somebody may contract the disease without having access to healthcare and then spread it even further. The World Health Organization and other global efforts are really being put into tamping down and treating all patients before it could potentially spread and become bigger, as what happened in the West African outbreak. And so just this morning, I think it was this morning, it might have been yesterday, the WHO released yet another update talking about who is receiving what different experimental treatments. So you can see that there is the MAB114 is in use, another potential experimental drug called remdesivir is in use, and ZMapp. And so currently 13 people have been given this trial antibody, MAB114. I think MAB114 only works against Ebola, Zaire Ebola virus. I don't believe that it is pan-Ebola virus, so it can be improved upon. At least 2 people who've been given this particular treatment have survived and recovered from Ebola. So let's talk just briefly about which type of experimental treatment would be ideal. So ZMapp needs to be kept frozen until it is injected or given intravenously over the course of a couple hours to patients who are being treated with it. So it's quite a labor-intensive and energy-intensive to keep frozen in a tropical area such as the Congo in order to transport it and make sure that it stays effective. Now, MAB114 can be refrigerated. It does have to be kept cold, and it's a much smaller injection, so it does not need to be administered with a one-on-one interaction with a healthcare worker in the same way. But ideally, of course, formulation would allow any type of protein or any type of therapy to be administered that does not need to be, or the therapy itself does not need to be kept refrigerated. And so the latest numbers with the Ebola outbreak that are ongoing, 120 cases have been confirmed molecularly. There have been 50 confirmed deaths, 30 probable deaths, so a total of 80. Sadly, a number of those who've been sickened have been healthcare workers, people who are trying to stem the continued spread of disease. 14 healthcare workers have been infected, and 1 has died of the disease so far. Let's hope that is something that we can say has come to a close by the next time we have Microbial Minutes. So just to reiterate what we talked about today, we talked about a foodborne bacteria that is linked to UTI causing E. coli. We talked about the bacterial tree of life and how it's been reorganized based on gene orders. We talked about broader HPV vaccination needed to stem cancer spread. And we discussed antibody, a new antibody that protects against multiple Ebola virus species. And fingers crossed that that is something that we are able to use in the near future. So there are a few questions. Thanks for your questions. I see Ivan has asked, does it affect if human clinical near the meat? Not 100% about the question. I'm guessing how you're talking about the E. coli question, and how does it affect if people are close to the meat. Now, most of those E. colis that are on the meat that you buy at the grocery store are going to be killed and gotten rid of by heating and properly cooking the meat to the right temperature. So that's not something that most people will have to worry about. It's usually undercooked meat, or the poor practice of washing one's hand or not washing one's hands after handling raw meat, right? It's very important not only to wash your hands, but to wash any surfaces or utensils or counter space on which those raw meat products have touched. And often it's not necessarily the meat itself, but some sort of contamination through transfer on non-sterile surfaces that can lead to that type of infection, and then incorporating the E. coli into one's microbiota. So my recommendation, and I hope, Ivan, that I'm answering your question, but my recommendation is that if you are buying meat to eat at a grocery store, having E. coli does not necessarily mean that it is the type of E. coli that will make you sick. The best thing that you can do is to cook that meat to temperatures that are prescribed. The USDA has a link, and we'll add that to at least the blog, if not the show notes page here. And then to wash everything very thoroughly with soap and water should be fine. And I also see that Eva has asked a question asking about wart treatments to clear the virus after boy is present. I think that this has to do with treating the warts themselves, which can be contagious. There's a lot of virus that is being produced actively in those warts. And I know that both genital warts and other warts that are caused by HPV, you can get them all over your body, very common in little kids, can be treated with salicylic acid, or even, I believe they can even be frozen off, and that will get rid of most of the active virus in that case. I don't know if that's what you're talking about, Eva, but I don't think that that those treatments have been able to as successfully ward off or prevent the warts from developing in the first place. And that's why the vaccination is such a great development, because it's not a therapy for people who have already developed the warts, but it's prevention from those warts from forming and being potentially contagious in the first place. I hope that answers your question. All right, thanks a bunch for the questions. Always enjoy being able to interact with you guys a little bit. I think that's going to wrap it for today's Microbial Minutes session, and we will be back soon with more hot things that have happened in the microbial sciences.